Okay, thank you for the organizer that having me today. So I'm very happy to be surrounded by people actually smarter than me. And I learned a lot from this morning and earlier this afternoon. So we're moving on, shifting the gear from non-clinical to clinical part. And so we're going to talk about the basic of clinical trial design. Okay. So, according to Professor Garmit, he told me to talk about some basic too, because some people here may not probably involved in preclinical part, okay, and probably not familiar with clinical area. So I'm going to talk about the overview and the basic of clinical trial design, like population, intervention, comparison, outcomes, and strategy to conduct successful clinical trial. Okay, and we've been talking about preclinical phase, okay, starting from animal model, and now we have a treatment, okay, that want to be tested in human. So there are 4 phases in human trial, starting from phase 1, 2, 3, and 4. So phase 1, the purpose is to find the maximum tolerated dose and to make sure the drug is safe, the treatment is safe, okay? So this begins with maybe healthy volunteers or patients, okay, in a small number of group of people that get tested, okay? Most of the time, starting from lowest dose, okay, that identified from animal model, and escalate the dose to find the maximum tolerated dose. So once the safety profile is established, at some point, okay, we shift to phase 2. So phase 2, the purpose is to find the efficacy, okay. So there will be expanded group of patients with the condition that we are interested, okay, maybe like about in 100 to 300 patients. So once, okay, this treatment is proven, okay, it seems to be effective, okay. Phase 3 will be expanded, okay, to 1,000 participants to compare with standard of care, to show the efficacy and also capture safety profile that could be missed in a smaller cohort. So after phase 3, so phase 3 is very important, okay, to get approved by FDA. So after the drug approved by FDA, it will enter phase 4. It's post-marketing. So this, they will continue to collect the data to capture the rare adverse effect that could happen. Okay. So this is traditional first in human study. This is like very basic 3+3 design. So this begins with maybe a healthy volunteer, a group of 3 patients. So the drug is administered to 3 volunteers. Okay. If there's no side effect occurs, they will move on to the next dose. But if there's a toxicity occurs, okay, in 1 out of 3 participants, okay, they will repeat the same dose again. If the toxicity occurs in 1 out of 6 participants, they will move to the next dose, okay. But if adverse event happens in more than 1 out of 6 participants, okay, they may declare this is the maximum tolerated dose or move to the lower dose. And if the first cohort, okay, adverse event occur in more than 1 out of 3 participants, they may do the same thing, declare this is a maximum tolerated dose or go to the lower dose. So this is from assumption that the toxicity increased with the dose in traditional way, the small molecule try, but probably not apply to cellular therapy. Okay, So once we have the safe dose, okay, we will go on to phase 2 and 3 clinical trial. So we have a group of patients with the condition of interest, right? And we want to compare that the new intervention is at least as effective as the standard of care. Okay, so the participant will be randomly assigned to intervention or control, which most of the time is standard of care. Okay, and follow up the patient and see whether the treatment is effective or having any side effects between the groups. So this is a very classic study design. Okay, I just want to show you. So we talk about clinical trial phases from phase 1 to phase 3, and phase 3 usually involves like thousands of patients. But I just want to show you the pivotal clinical trial for ATMP approved in EU. So most of them, they got approved in early phase, like phase 2, okay? And it's not randomized, only single arm, okay? There is no control, or some of them use historical control. And the endpoints, they use intermediate endpoints, not a hard endpoint, okay? And you see the number of patients, enrolled patients, like just a few hundred patients, so much lower than traditional trials. Why is that? Okay. I think the issue is like most of them are orphan disease. There is no treatment available, and there's a lot of pressure, okay, to get this treatment out to the market. And also to deliver this treatment, also complicated and expensive. Also, so there are some limitations out there, so that's why like they got approved at early phase with fewer number of patients. Okay, sorry, it's, so you can see the success rate by FDA. Okay, for our medicine combined, okay, the success rate is about 76%. For ATMPs overall, is lower to 59%. Okay, and for non-orphan ATMP, the success rate go down to just 50%. Okay, and the reason for failure is insufficient efficacy demonstration, and that they use non-validated primary endpoints, and efficacy claims by non-pre-specified post-hoc analysis, not the primary endpoint that's planned, okay? And limited safety database. And also there's some risk with immunogenicity. As have been pointed out earlier, so CGT trial is not straightforward as small molecules that we're very familiar with. So we a lack of experience, and this type of treatment just come out not really long. So the long-term effect is not known. And like from earlier, Professor Kevin pointed out there's some persistence of the treatment also. So what that treatment would do and react to our body is not known. There's some report of lymphoma after CAR T-cell treatment also that we have to be aware. And treatment may involve very invasive procedures to deliver to the target site. Okay. So the objectives of the trial to prove, to investigate the efficacy and safety of CGT will be involve 5 areas. So the first one is dose exploration for early phase. Second one is activity assessment, safety assessment, efficacy, and feasibility. Because to deliver the treatment is very complicated procedure. Okay, we have to assess this if this is out to the market, would that apply? Would be applicable to the public at all? Okay. So about the basic of the design. The first one is population. As I mentioned earlier, so usually like first in human trial, usually enroll healthy volunteers, but may not apply to CGT at all because high risk associated with treatment. So, so most of the time, phase 1 study will enroll like the patient with target condition, okay? And it is also a hard choice, okay? We want to treat this disease, but at what stage, okay? If we select the patient with later stage, okay, and they may have no other available therapies, they may accept more risk. But also, they may not tolerate the procedure that well, right? So there's some risk and benefit out there that you have to consider. And also, we have to consider about manufacturing failure. Let's say for CAR T cell, we have to do a cell harvest, whether the patient can tolerate that at all. For example, okay, we doing a CAR T cell therapy in patient with SLE, autoimmune disease. This patient, they have autoimmune and have inflammatory condition in their organs. Okay, and they need to get immunosuppressive therapy to control that disease. But when we're doing leukapheresis, the cell harvest, to produce CAR T cell, we have to stop immunosuppressive treatment. Otherwise, it will interfere with production. So this is very critical to find an optimal balance. Okay, when we stop the medication, and if the patient can, like, how long can the patient can go on without the therapy to collect the cell. And next is the patient may meet entry criteria at screening, but their condition may deteriorate, and they cannot accept the therapy anymore once the product is completed. Okay, I will give an example again. My patient with SLE, they have, I'm a nephrology doctor, so I'm a kidney doctor. So they have declining kidney function because the disease is not very hard, it's very hard to control. Okay, while we're waiting for the product to complete, their kidney function continues to drop. Okay, and if it's lower than certain point, they cannot have chemotherapy before getting CAR T cell. So we're competing with the time. So when you plan to enroll the patient or write a protocol, you have to consider this factor too. It's not like what we have done before. Okay, it's more complicated. And the cohort size, usually small. Like we begin with like maybe 3 to 6 patients because there is associated with high risk, but you may consider a larger cohort if the risk is not as tolerant. Okay. And next is intervention. So first, in human trial, it's very difficult to find the right dose because it's not as straightforward as small molecule study that we have like some established model to predict, like translate from animal model to human. But like in cell therapy, like what's the good dose? Okay, and also if the therapy consists of like many cell types, okay, if therapy effect may be related to one cell type, but side effect may be related to the other. And how you escalate the regimen, okay, when you step up the dose. This is what I've learned from FDA. So usually increase in half log, okay, I'm not sure that I'm right, but this is what I learned from the FDA. So about like 3-fold increment. But the interval between the dose who knows? Because like the small molecule, we know pharmacokinetics. Okay, we can wait until like the drug is like out of the system. But for cellular therapy, what is the optimum window? Okay, to see acute or subacute adverse event, because you see the graph earlier by Dr. Gavin, like the dynamic of the cells varies too. Okay. And the intervention to deliver the treatment involve a lot of complex process. Okay, you need to have a standard established SOP to ensure that everything is consistent and safe for the patient. And you also have to think about what if there's a manufacturing failure, you cannot make the product. Okay, what would you do next? Okay, would you retreat the patient again? What would you do? And you also have to make sure that the patient will not get high-risk therapy, like lymphodepletion therapy, until you know that CAR-T product is finished and good to give to patients. Okay. And what about the patient cannot receive the treatment? Okay, we have to have a plan B. Okay, whether you're going to replace untreated subject by increasing enrollment or not. Okay, so if we give intervention, actually most of the time we want to compare with if the other group that gets standard of care, but probably not a case of CGT too. It's very hard to find control in certain conditions. So how do you compare the efficacy? Ideally, you can have concurrent control group getting another types of therapy. But if not, okay, we can do historical control, okay, by getting the data from retrospective data or the data from completed trial or registry. Okay. There are also sometime there will be a master protocol that treat the same disease with a single protocol and use a common control arm. Okay. And typically we do blinding in RCT, right? But in CGT, probably not a case too. This is too risky and complicated to do. Okay, for the next one is outcomes. So we have to think about to assess the activity, safety, and efficacy and feasibility. So it depends on what you're looking at. Okay, we may measure gene expression, cell engraftment, morphologic alternation, immune function, tumor shrink rate, and various stuff. But this is kind of like you have to think broadly because we don't know the consequence of this therapy. So please consider like saving some biobanks for future analysis too. Okay. And the outcome. So to determine the efficacy of the treatment, sometimes we follow up the patient for many years and look at the hard outcome like mortality or disease-free survival. But probably it's not a case that we need the therapy for the patient that doesn't have any therapy available for them. So sometimes we have to use a surrogate endpoint. There is a table in FDA that they recommend surrogate endpoints for some disease. You may consider like looking at that table as well. For someone not familiar with this, what is a surrogate endpoint? So let's say I'm a kidney doctor, okay? What we would like to know at the end, with this therapy, would that prevent kidney failure or not? Okay, but I have to follow up the patient for maybe 10 years to meet that outcome. So we use a surrogate endpoint like the decrease in kidney function, for example. Okay. And about safety, we want to capture anything related to the treatment, like AE related to the procedure, like leukapheresis or lymphodepletion therapy, like by giving chemotherapy, would that cause any unnecessary side effect compared to standard therapy? Or adverse event related to cellular or gene therapy, like inflammatory reaction or neurotoxicity or graft versus host disease, and etc. That we have to monitor that. And at least 1 year monitoring should be done, okay, or longer to detect any unknown side effect that could happen later. And you should also put in a stopping rule, okay, for individual stopping criteria, like when you are going to pause the treatment for individual patient, or the stopping criteria whether you're gonna proceed to the next dose or not, or what is the criteria to terminate the trial. Okay, and most of the time, IRB would recommend data safety monitoring board. Okay, what are they? So they are a party of experts that is independent of the study team. Okay, they consist of clinical experts in that area, biostatistician, and investigators who are with expertise with clinical trial design. Okay. So they go in to review the data in case of some safety concerns or some deviation, and they have a power to judge whether the study should be terminated or modified. And something that kind of like not commonly done in traditional clinical trial is feasibility assessment. So we have to analyze the proportion of patients who fail to receive the treatment too, and to identify the factor that associated with treatment failure. Okay. So these are kind of basic that to cover like what you should consider when planning to do a clinical trial in human related to CGT, and Professor Goramit will kind of like fill in later. Okay. And the strategy to conduct the trial, I mean, like we learn to be a scientist and to carry on like the trial successfully, like need more skill, like marketing. You have to be a team leader, could be a labor sometime, and know about finance and how to run everything, put everything together and run it. Okay. So a lot of things you have to manage. Okay, you have a research question. We were just talking about the proposal part, but there are a lot of things involved. You have to apply for a grant, okay? And this is very expensive to conduct a trial related to CGT, and you have to convince IRB, okay, to approve your study. Sometimes it takes, like Ajahn Goramit, like the first time, it took him like a year to get approval, okay? But it's faster for us after like he paved the way. So you have to consider the timing too. You have to do IRB application, clinical trial registration, and the logistic of the budget too. Okay, how much where you can apply for, how much, and when the money will come. Okay. When you be able to utilize the money, that's a factor too. You have to think about the recruitment plan, data management, clinical workflow is very important, okay? Because it's very risky procedure. It's not only study team, but other specialists should involve, like hematologists, infectious doctor, or ICU doctors. Okay, we have to need a good facility to handle this therapy as well. Plan about the lab, whether you're gonna have save the biobank, medication, finance. As I told you, it is very expensive to conduct a trial for CGT. So for budget calculation, we usually break down the activity into the visit, okay, and see what we need to spend, okay. But there are other costs too associated with the trial that you should consider. Most of the time we have to get clinical trial insurance, okay. This costs a lot. Also, you have to think about data management. In case you will have FDA come and look at your data, you have a good, solid data management plan, okay, and have audit trail for them to trace. Study coordination, lab logistic, and about the intervention and other stuff. And you may need to have CRA to monitor your study as well. And this is like an example of clinical trial insurance we got, okay. Like for this, you have to look at the coverage, okay, of the insurance. Also, the duration, look at the quotation that they give you. And also sometimes they talk about deductible, okay. What is deductible? Is this when you want to claim, okay, if you have an issue and you want to claim for reimbursement, deductible is the amount that you have to pay, okay, to get reimbursement. So you have to consider about that and have some savings for that as well, okay. So this is like the resource that I use, and I think that's all I have for now for the basic. Thank you.
Thank you, Dr. Wonngarm, for her talk, and the door is open for the question.
Thank you so much for very comprehensive talk. So about the study design for phase 1. So mostly we use like conventional 3+3 trial, right, for conventional trial. Like for CGT, you have any recommendation for like adaptive design or any other approach that we can use other than like rule-based approach that we use in conventional study?
Okay, I think you're gonna talk about that later, right? Okay, I'm sorry, but yeah, like you mentioned, so the traditional design is really fixed, okay, and actually we don't know what we're gonna find out when we conduct the study, okay. So let's say if you want to like pre-specify whether they're gonna give this dose and this dose and this dose, but it doesn't happen that way. Okay, maybe we need to modify to give a lower dose, a higher dose, or enroll more patients. This is like Ajahn Goramit talk mentioned that this is adaptive trial design that kind of like more adopt nowadays. It will be more flexible. Okay, you can, but this should be planned ahead. If you want to, how you want to modify, okay, you have to think about what could happen, like different scenario that could happen. Like if this happened, like, okay, if this happened, case A, we're gonna do this. Case B, we're gonna do this. This is like kind of adaptive design, but this involves like more sophisticated statistical analysis as well. So you need to discuss with statistician and probably like FDA if you plan this.
Thank you very much for your talk. And I still have a very basic question regarding the trial. So I'm wondering if, as a follow-up question to what you just said about, sometimes we have to adapt as we go along, right? But wouldn't that consider deviation from the IRB? Like if we're about to do something that's different from what we propose in the IRB, like, yeah, what?
Yeah, so that's why it's like should plan ahead. This should be included in the protocol, in the adaptive design. But it's still like it's flexible, but should be still written out beforehand. Okay, like for example, like I've done one adaptive design. Okay, we written like there's a written condition if like higher dose show efficacy, we may add another higher dose. Okay, but the lower dose show more efficacy. Like if the high dose is not safe enough, like the lower dose we may add another arm, just to make sure like the lower dose, like which one is the best or something like that. But yeah, you have to plan ahead.
I see. Thank you very much. So pretty much like plan ahead and write everything down in the IRB like before. Thank you.